Quorum means sufficient numbers, but what does it have to do with prokaryotes? Well, just like how we communicate with others through text messaging, bacteria also have a form of communication. Turns out they communicate between different species and not just the same species. They use regulatory pathways controlled by the density of cells of their own kind to regulate many functions. It's a mechanism to assess population density. With this approach, they can ensure that sufficient cell numbers are present before initiating activities that require a certain cell density to work effectively. Take the production of toxins, for example. It does not make sense for one of them to produce some, when instead, if large number of cells strike at the same time, they will more likely to achieve whatever they intend to do. This phenomenon was discovered as a mechanism by which light emission in bioluminescent bacteria is regulated, including the marine bacterium Alivibrio fischeri. The bobtail squid lives in symbiosis with this bacteria, which are capable of emitting fluorescence generated by the enzyme luciferase. The lux operon encodes the proteins needed for bioluminescence. However, this bacteria by themselves do not emit light, but when all together in the gut, emits light. So, during the day the squid hides, but at night they come out to hunt. During the day, Alivibrio fischeri starts colonizing the gut, reaches the gut, and then loses its ability to move and stays in the organ. At night, the gut emits fluorescence, making the squid camouflage against the moonlight and stars. In the morning, they vent all the bacteria back out into the seawater. Now, time to learn how this mechanism really works in detail.
Each species of bacterium that employs quorum sensing synthesizes a specific signal molecule called an autoinducer. This molecule usually diffuses freely across the cell envelope in either direction. Because of this, the autoinducer reaches high concentration inside the cell only if there are many cells nearby, each making the same autoinducer. Inside the cell, the autoinducer binds to a specific transcriptional activator protein or a sensor kinase of a two-component system, ultimately triggering transcription of a specific genes. While several different classes of autoinducer exist, the first to be identified were the alpha-homoserine lactone, the AHLs. They are under control of the activator protein LuxR and are induced when the concentration of the specific A. fischeri AHL becomes high enough. This AHL is synthesized by the enzyme encoded by LuxI gene.
The lux operon is an inducible operon similar to the lac operon. The lux operon consists of LuxR, the promoter, LuxI, LuxC, D, A, B, E, and G. LuxR generates the LuxR regulator, which will be discussed later, and the promoter is what activates the operon. LuxI produces the LuxI enzyme. LuxC, D, E, and G create other polypeptides related to luciferase, while LuxA and B generate the alpha and beta subunits of luciferase, respectively. There is always a basal amount of expression of LuxI, and so in any cell at any time, there will always be some small amount of LuxI enzyme. What the LuxI enzyme does is it takes substrates for AHL, which are floating around the cell, and converts them into active AHL. AHL can then diffuse out of the cell. This is just a random estimate, but imagine that one cell produces 5 molecules of AHL. This is a small amount, and so there is not very much force for AHL to move to diffuse back into the cell once it has diffused out. However, imagine there were 20 cells all next to each other. The total amount of AHL produced would be 100. This is a much larger amount of molecules in the extracellular space, and so there would be a larger force for diffusion back into the cell.
When there is a high amount of AHL inside the cell, the AHL can bind to the activator, the LuxR regulator, which is represented in blue. The LuxR regulator's actions will be further discussed in the next section.
The next part is the binding of the RNA polymerase on the gene of the bacterium. RNA polymerase will bind to the LOX operon at a site of the promoter region. Once bound, RNA polymerase will transcribe the entire LOX operon. The LOX operon has 5 genes that would be transcribed total. Of those 5 genes, the 2 main gene we'll focus on is LuxA and LuxB. Once combined, LuxA and B will form a protein that is able to promote light in the bacterium.